Today we have the pleasure to host Cyril Poupon. So Cyril is a serial researcher working at Neurospin. Actually, he has been working at Neurospin since Neurospin opened, and he was already at the serial before that. Cyril specialized in various aspects of MRI physics, and in particular, diffusion imaging, DTI. And today he's going to present a very ambitious project about massive scanning of a small piece of cortex. So thank you very much, Cyril, for your presentation today.
Thank you, Florent, and thank you to the audience, and thank you to give me the opportunity to make this presentation. So, first of all, I would like to say that this project, which is a kind of massive scanning project, is of course something that was done in the frame of a team, which is the GINCO team, which is a team who is focused on diffusion MRI and quantitative MRI, and it's the GINCO team, and it's part of the Baobab research unit of Neurospin. And so the title is Chenonceau, a mesoscopic ultra-high field diffusion MRI mapping of the human-brain connectivity at 11.7 Tesla. So I want first to say that it's not in vivo. That's not on the big 11.7 Tesla of Neurospin. It's on a random 11.7 Tesla preclinical MRI system. And it's post-mortem. So now that I've said that, I have prepared, of course, a small outline where after the introduction, I don't know, most of you are familiar with diffusion MRI, but I will just give again, the very basic physical principle of diffusion MRI. And then I will try to explain why we wanted to go on a post-mortem diffusion MRI and what the kind of resolution we can expect from diffusion MRI. And before presenting the whole Chenonceau project and the different steps to achieve what we have now, we had to do some proof of concepts, and we first started with an hippocampus, a human hippocampus, and then with a human brainstem. And then at the end, I will provide some perspective and conclusion of what we've done. So this is a slide that was made by Jean-François Mangin. And this project is somewhat part of the Human Brain Project, but also part of another ANR project in close collaboration with my collaborators at Tours that I will mention right after. And the idea was to try to look at the connectivity, but at various scales. We all know that the human brain can be considered at various scales. And of course, it's the same for the connectivity. And here I want to mention that's the structural connectivity. I'm not talking about the functional connectivity, even though the two are related by this notion of connectome that was introduced now more than 15 years ago by Hagman and Sporn in 2005. And this connectivity in the frame of the Human Brain Project has been tackled at 3 different scales. So some of our colleagues are doing histology and microscopy at the microscopic scale using 2-photon fluorescent microscopy or immunohistochemistry. And that is done both in Italy and in Amsterdam. And at the very end, we have the millimeter scale with several people working on it. Jeff establishing the atlas of white matter bundles, and also some German colleagues and UK colleagues. And this is also related. We have the opportunity, to try to make some comparison between what we have established using diffusion MRI in terms of superficial connectivity, with what can be observed using IEEG, and that is done by the team of Olivier David in Grenoble and Marseille. And as for us, for the intermediate scales, we have 2 modalities. One of the modality is polarized imaging. That is one of the key modality developed by by the Forschungszentrum Jülich, and in particular by Markus Axer, where he can scanned some histological slices at the microscale in plane, but 500 micrometer in plane. And from those high resolution imaging, it can reconstruct, maps of local orientations at 50 micrometers isotropic scale. And we propose to do another scan using a whole brain postmortem, whole postmortem human brain, using ultra-high field magnetic resonance imaging, but also strong gradients. And we are here where we wanted to scan at a mesoscopic resolution between 100 and 300 micrometers. And that will be the main topic, of the presentation today. But before starting, for those who are familiar with diffusion MRI, I just want to go on a recent publication by Yendiki, who is based at the MGH, and who recently wrote a bioarchive paper entitled Post-mortem Mapping of Connectional Anatomy for the Validation of Diffusion MRI. And the purpose was to try to evaluate the accuracy of diffusion MRI orientation estimates obtained with different Q-space sampling schemes. We have many, many ways, and many models to infer the local orientation from which we can do tractography. And it is not clear what are the key factors. And in fact, the conclusion, of this very interesting paper was that, in fact, not only the angular resolution of the orientational profile we acquire is important, but the spatial resolution emerge as a key factor. So it's not because you will increase the number of directions that you will scan over a sphere that you will enhance the resolution of your tractograms. Neither is the enhancements of your b-value. We all know that the b-value is an important key factor to get high resolution angular profiles. And more people, and I was part of those people, thought that it is sufficient. It's not interesting to have a very high spatial resolution. It's sufficient to get a very high b-value, because within a single voxel, you can get a very rich information about the various populations crossing themselves within the voxel. But in fact, no. The spatial resolution plays a key factor, because the kind of configurations we can find within the voxel are so complex that it's better to combine both high angular resolution with high spatial resolution. And that was one of the reasons why we wanted to scan a single human postmortem brain at a very high resolution. And this is another paper, just making a summary of what you can find in terms of post-mortem imaging, post-mortem diffusion MRI imaging. And this was taken from the last presentation of my PhD student, Alexandros Popov, who did a presentation at the ISMRM in 2021. And in fact, you have here the scale of the resolution. And as you can see, the first post-mortem human brain scan using diffusion MRI were done at the millimeter resolution. And it was first done, in 2014, with a very high b-value and a moderate number of diffusion directions. And a little bit later, there was the introduction of BSSFP sequences, in particular by the team of Carla Miller, where she could reach a better resolution of 0.73 millimeter isotropic resolution, still with a high b-value, but still moderate number of diffusion directions. And there was not so many work to go beyond. There was one work by Robin and Duyman at 7 Tesla, where he could get a similar spatial resolution that Carla Miller obtained in vivo, but at a really moderate b-value of 10 hundredths per millimeter square. And so the result we got here is a kind of jump, because we could go up to a 0.2 millimeter isotropic resolution And we had the opportunity to have very different B values and up to 8 seconds per millimeter square, and with a quite high number of diffusion directions. So we feel that we have somewhat, provided a new kind of diffusion MRI data set that would worth being investigated to see whether it can enhance the view on the structural connectome of the human brain we can have access to today. So now I go to the principle of diffusion MRI, and I like this picture. I mean, diffusion MRI is a very basic principle. If you inject a bubble of ink in a glass of water, and if you wait for a certain amount of time that we call the diffusion time, you will see that the ink will spread over the entire glass, and at the end you will have an homogeneous blue color, which is corresponding to the diffusion MRI process within an unrestricted media. And there is only one equation corresponding to this process, which is the diffusion or heat equation, that tells you that the temporal derivative of the probability distribution function of the displacement of water molecules is related to the Laplacian, to the spatial Laplacian of this probability distribution function. And the link between the two is this famous diffusion coefficient D of the second Fick law, and R is the displacement vector, and this equation just tells you that if you put a set of molecules at the center of this frame at a starting time, and if you wait, let's say, for 100 milliseconds, at the end, the water molecules will have those trajectories represented in red here, and their position is represented after these 100 milliseconds in green here, and there is the same chance to be in any orientation of the 3D space. So why it is interesting for probing the tissue orientation? I mean, if we now consider white matter, so white matter, here you have a slice representing the corpus callosum, which is made of myelinated axons, but it would be true also for unmyelinated axons. In fact, if you're looking at the water moving, and populating, the brain, the water present in the axons will have a non-isotropic displacement pattern, like it is represented here. If you put the same amount of water molecules at the center on the axis of the axon, and if you are waiting for a certain amount of time, then you will observe that it is easier for the water molecules to move along the axis of the axon than along any direction perpendicular to the axon, because of the restriction imposed by the membrane of the axons. And so this is a very interesting way to get access to the local orientation of the human connections, because if you are able to probe the motion of water molecules, then the main directions of the orientation distribution functions of the movement of those molecules gives you the orientation of the axons. So that's the basic principle. And of course, how to be sensitive to this motion in using an MRI system. In fact, we are using some strong gradients that just label the position of the spins, and if you then take a photo between two different instants of the position of the spins, you can, go back to the information that provides you the local orientation of the axons. And there exists, in fact, plethora of acquisition schemes, and there is a link between the space that we want to measure, which is the space of the probability distribution functions of the displacement of water molecules, and another space which is related to what we measure in physics, which is the Q space. And the Q space is just like a Fourier transform of the displacement space. and there is a Fourier relationship between the two spaces, which means that in addition to have a first Fourier transform between the K space we are measuring in classical MRI and the volume space that we want to reconstruct. Within each voxel, there is another spherical Fourier transform that makes the link between some measurement in that Q space and the propagator, I mean the probability of displacement of water molecules within that voxel. And typically what we do, we acquire some diffusion weighted volumes along many orientations. So you can see that there are many green spots on the three spheres that you can observe in this slide. And each spot corresponds to a specific diffusion orientations plus to a specific B-value. And in general, what we do, we collect 1, 2 to 3 different spheres in that Q space with a number of orientations that generally increases when we go from the lowest B-values to the highest B-value. And this is our input data. And there is a modification of the Bloch equation, so I won't comment on, of course, the equations, but there is a modification of the equation, the Bloch equation that typically drives the magnetization of the spins in class conventional MRI. That was done by Torre, which add a further term related to the diffusion process that just tell you that the temporal variation of the magnetization is related to the Laplacian of this magnetization, spatial Laplacian of this magnetization related to the diffusion coefficients. And if you look at those two terms, you should remember that, I mean, the propagator is driven by a similar equation with the propagator. So in fact, the magnetization is summated to exactly the same heat equation than the propagator of the displacement of water molecules. So you will find a diffusion equation in the magnetization that allows you, in fact, at the end, to measure a signal which corresponds to a magnetization, which is related to a conventional T2 weighted signal, plus an additional decay corresponding to the diffusion decay. And what you can observe here is that the signal for a given point in the Q space corresponds to an attenuation of the T2 weighted signal related to the diffusion coefficient along this orientation times a second parameter, which is in fact nothing more than the B value. So just to summarize, when you are doing diffusion imaging, you are only measuring an attenuation of the signal, which is related to the diffusion of the water in the orientation corresponding to the Q space you have acquired in your Q space, to the Q space point that you have acquired in your Q space. And from that equation, you can, and from a series of acquisitions that you can do, you can model an important function for us, which is the orientation distribution function, which provides the main orientation of local tissue. And there exists a plethora of models, but I would mention only the most popular ones and the most used ones. So the historical one was the diffusion tensor imaging model. Then came the Q-ball model, and now, I mean, people more and more use the multi-shell, multi-tissue constrained spherical deconvolution model, which at the end provide you here, you have a picture of the color encoded orientation of the tissue. So the red axons are along the x-axis of the slide. The blue axons are aligned in the direction in the y-axis of the slide, and I mean the The axons oriented perpendicular to the slides are represented in green here. And for each voxel, you can get access to a 3D orientation probability, where the main lobes of those orientation probability are the putatively corresponding main orientations of the underlying axons. And then what you do, you are doing fiber tracking. So because you have access to the entire map of the entire brain, on the entire brain of the orientation distribution functions present within each voxel, what you do, you're just following the main, the most the most probable, pathways to infinitely reconstruct such kind of connectograms. Connectograms being a set of virtual connections connecting different regions of the brains. Okay, and here again there are a plethora of methods to do that, from deterministic to probabilistic to Bayesian to global approaches. So I don't have time to enter into the details, but here again there is another step of modeling. And what the only thing I would like to say is that, I mean, reconstructing the fibers from this kind of map of orientation distribution functions is an ill-posed problem by nature. And so we have to develop more and more complex stratigraphy algorithm to embed some anatomical priors or some microstructural priors in order to reduce the number of false positives that are generated by the stratigraphy methods. So now I would like to say why doing post-mortem diffusion MRI, and I'd like to have a discussion about what is the best imaging system to do that. Shall we use a small animal or a human MRI system? And to do that, I just reproduced here a table where we have tried to establish the pros and cons for the two different. So first, the lines here correspond to the first line, correspond to the in vivo acquisition versus ex vivo acquisition, and here clinical versus preclinical MRI system to perform your acquisitions. So if I consider an in vivo scanning of the brain, of course, the main severe time limitation is the 1.5 hours scan duration limit that we've got. We cannot put the human subject for a long time in the magnet for ethical reason, and I fully agree with that. The second point is that when you are scanning in vivo, there is a lot of motion artifact that can occur during the acquisition, which will somewhat corrupt the image quality. And when you are doing in vivo acquisition, you cannot do any histological validations. If some of you would like to give their brain to the science, I'm open to scan their brain before and after death, of course. And the good news for in vivo imaging, it's of course, that's the best configuration to preserve the tissue integrity. The tissue are living, so the results measure something that is really happening in the life. And you can add to the diffusion MRI acquisition a bunch of functional MRI scans to make some correlation between structural and functional connectivity. Now, if you are looking at the same situation for ex vivo imaging, of course, the main and most interesting point is that there is no time limitation. So you can theoretically reach higher SNR and you will be able to enhance your spatial resolution. The dead brain never moves, so you don't have any motion artifacts. So it also somewhat contributes to enhance the image quality. And you can, of course, do some validation using immunohistochemistry, which is the main point here, that you will be able to correlate with various modalities what you observed. But there is a risk that you do not preserve the integrity of the tissue. So there is an important point that should be taken into account. It's the preparation of the brain and the preservation of your brain. And naturally, this is not possible to do functional MRI, even so some nice people have produced some functional MRI activation maps on salmons in the past. But, well, you don't have, I mean, the correlation with functional MRI. And now, if I want to do ex vivo MRI, should I use a clinical or a preclinical system? Both the system can benefit from ultra-high field magnets. So this will contribute to enhance your spatial resolution. But I would say that with preclinical system, you can even reach higher magnetic fields. Here at Neurospin, we can go up to 17 Tesla. But the main difference concerns the gradient systems. The gradient systems in clinical systems, up to now, they are much more limited than what we can get in preclinical system. And there is a factor of 10 between the power and the maximum gradient magnitude you can reach with a clinical system versus a preclinical system. Today at Neurospin, we can go up to 100 millitesla per meter on our 70, but we can go up to 1000 millitesla per meter with our 70T system. So this is very important because we are doing diffusion MRI where the gradient pulses are used to encode the position of spins. The higher the gradient magnitude, the more precise the position of spins you get. So it will contribute to enhance your angular resolution or the resolution of your propagator. Another point that we consider less in general is the antenna. When you have a clinical system, the loops to collect the signal from your antenna, they are generally far from most of the voxels populating your brain except the cortex, which is a nice information when you are doing functional imaging because the activation are located on the cortex or they are close to the loop antenna, to the antenna loops, sorry. But this is not the case for the connectivity, which can be deeply located inside the brain. So if you are doing, diffusion MRI with a preclinical MRI system, then you will ensure that all the voxels remains close to the antenna loops. So in theory, you will get a higher SNR with the combination of preclinical imaging and the small antenna that you are using. Of course, the benefit of a clinical system is that it can contain an entire human brain, which is not the case of the preclinical system, and the brain has to be cut. So that's something that should be carefully taken into account. But now, if you summarize, you will clearly see that first, doing ex vivo imaging on the human brain for diffusion MRI is more promising using a preclinical system than with using a clinical system. And so if I look at the systems available at Neurospin, we have two clinical systems. We will soon have a third one at 11.7 Tesla, and we have three preclinical systems. And if you are trying to expect what can be the resolution that you can achieve with those systems, in vivo, typically you are using a PGAC PI2D sequence, and so you will be able to reach a resolution up to 1.2 to 2 millimeter isotropic. It depends also on the scan duration that is allowed by the clinicians for a given protocol. Ex vivo, we have a balanced SSFP sequence that I've brought from the NGH, and you can go up to 0.4 to 1 millimeter resolution. Okay, so it's better. And now, if we want to do the same thing on the preclinical system, if you are using a PGAC EPI 2D sequence, then the resolution can be can be pushed up to 0.25, 0.40 millimeter when you are scanning in vivo, because I mean you have to keep your scan duration below, I would say, a couple to 6 hours maximum with anesthetized animals. And if you are doing ex vivo imaging, then you can use 3D EPI PGSE sequences, and in that case you can go up to 0.1 millimeter isotropic on the 17T and to reasonably 0.2 millimeter isotropic on the 11.7T. And as you can see here, we have chosen to use the 11.7T because, I mean, the PharmaScan was too old at the time we started the project, and because, I mean, we did not get convenient antenna on the 17T, but I must admit that now we could probably use, I mean, the 17T to do that. I mentioned earlier that the tissue preparations are very important because, I mean, ex vivo, there are a lot of tissue modifications. And I would like to highlight the importance of the type and the quality of the tissue fixation. The fixation should not be done only by immersion in PFA, because in that situation, if you just put your brain into formaldehyde, then you will have a nice fixation of the cortical surface, but the deepest tissue areas are less well fixated. So what our colleagues from Tours did, I mean, they collect the brain from people having given their body to science, and then they inject PFA through the arteries, which ensure a very stable fixation and a constant fixation deep inside the brain and also at the surface of the brain. And if you do that, it also helps you removing the blood from arteries and from the vasculature of the brain. So you will, at the end, also have less susceptibility artifacts, which is important when you want to do EPI, even though, I mean, you would use segmented EPI sequences. And of course, the fixation induces a loss of T2. So your acquisition window will be much shorter than the one you could reach with in vivo imaging. So you will have to get strong gradients to somewhat tackle this problem. And another point in that when you do the fixation, the brain should be put and maintained in sustentation so that, I mean, the brain does not take the shape of your container, which is somewhat you quite often see people having a really cylindrical brain. It's just because, I mean, it took the shape of the brain during fixation. After you have done the fixation, you have another step to be done. It's the radiation of your brain in order to recover partly T1 and T2 relaxation times. So the fixation, it should, I mean, for the fixation, it should last at least 4 to 6 months for a human brain. And for the irradiation process, you should at least irradiate your brain for 2 months to partially recover and optimally recover the T1 and T2 relaxation times. But the T1 and T2 relaxation times still remains reduced. So as I mentioned, you have shorter acquisition windows, so you need to use strong gradient magnitudes to scan with fast acquisitions. Another point for diffusion is that the diffusivity ex vivo is reduced by a factor between 3 to 4 with respect to what you can get in vivo. So you need to use much larger B values. So it means either you need to use much stronger gradients. I mean, you need to use much stronger gradients. Why? Because if you want to increase the B value, either you have to extend the duration of your diffusion pulses, which is not good because at the same time, the T2 is diminished, is decreased. So you have to compensate that by the use of very high gradient magnitudes. And the last point is that the diffusivity depends on the temperature too. So you need to establish a protocol that ensure the preservation of the temperature during the acquisition that can last a few days. So you have to take care about all those features to have a nice acquisition. And so now I come to the first proof of concept that was done by Justine Beaujoin, who was a former PhD student of the team. And we got a temporal lobe, a piece of temporal lobe from the Jülich Focus Centrum team. And it was, this is a picture of that temporal lobe, including, the hippocampus. And we first developed a first protocol where we scan, this hippocampus, targeting targeting a T2-weighted spin echo anatomy and a 2D post-gradient spin echo diffusion weighting scan. And so for the anatomy, first we acquired, a scan at 200 micrometer isotropic. So the scan duration was 11 hours. And we, at that time, did a 2D PGSE segmented EPI spin echo sequence, and the target resolution was 300 micrometer isotropic. And here you have a picture of a diffusion weighted scan at a b-value of 4,500 seconds per millimeter squared, which shows you that there is quite a lot of signal. This is a diffusion weighted scan, this scan, so you have a lot of signal. And so we collected we collected 3 spheres at 3B value at 4,500, 7,500, and 10,000 seconds per millimeter square along various diffusion direction, and the scan duration was 12 days. And this is a picture of the anatomical scan that allowed Justine Beaujoin, to do the segmentation of the various subfields of the hippocampus, which at the end gives you this representation. And we could nicely, get a protocol to do the segmentation, and it was published in Brain Structure and Function. But it also allowed us to look at the connectivity, the inner connectivity of the hippocampus. So we just use the diffusion MRI data, and we computed some ODF maps from a tubal model, and then we performed some stratigraphy. So here you have an overlay of the surface rendering of the subfield of the hippocampus, plus the connectograms made up of lots of virtual connections. And we were able to efficiently reconstruct the mossy fibers, which are some fibers crossing here, the 2 subfields of the hippocampus, and also the perforant pathways as it was described in the Atlas of Duvernois. And I don't know if all of you knows, but Duvernois died a few weeks ago.
Cyril, excuse me to interrupt, but I'm looking a bit anxiously at the clock and your outline. So in principle, you have 10 minutes left. So, perhaps you can consider prioritizing the Chenonceau project.
Okay. I thought I had 1 hour, sorry. So at the end, what you can get to, you can establish a connectivity matrix of the subfields of the hippocampus, where we could clearly see, that the head of the hippocampus has more connection than the tail of the hippocampus, which is something which is known, of course. So now I will switch to the Chenonceau, if you want to, that we go to Chenonceau. So I just show you some nice pictures of the brainstem. And so the genesis of the Chenonceau project was in perfect line with this proof of concept, because we were successful in scanning a first sample, the hippocampus that I showed you previously. We now decided to scan an entire human brain, and we call it Chenonceau because, it was coming from the Faculty of Medicine of Tours, and it was prepared by Christophe Destrivieux, which is part of the INSERM iBrain research team. And it was corresponding to a 92-year-old male. It was fixed with the procedure I mentioned, through the arteries and plus immersion. and the brain was collecting only 8 hours after the death, so we could get access to the brain while it was, I would say, between brackets, still fresh, and there was no history of neurological disease. And as I mentioned, we had to prepare the sample, so the sample was first cut into its two hemispheres. The brainstem and the cerebellum was also collected and put apart. And each hemisphere was put in a gel to maintain, the brain and to enable reproducible scanning of the blocks that we have cut afterwards. So here you will see that each hemisphere was cut into thick layers, and after each thick layer was cut into blocks. And at the end, we have here 7 blocks or 6 blocks, depending on the hemisphere, put in a specific container that was designed by Jérémy. And then it was maintained and in a reproducible position with this system in our MRI system, and it is preserved in the fridge at 5 degrees and regularly radiated. And so in order to ensure the possibility to adequately reconstruct the brain, what we first did, was to scan, first the entire brain before cutting it on the Prisma system at a resolution of 500 micrometers, so using a T2 weighted space sequence. And then we also scanned on the 3T system the 7 blocks that we cut in order to have the possibility to do the restoration from each block to the brain and from the different field of views that we will acquire at 11.7T to each block. So we will have a 2-step restoration process. And to facilitate registration, each block was divided into 4 field of views with an overlap between each field of view, as you can see here. And then we went to scan the various field of views of all the blocks of the 2 hemispheres at 11.7 Tesla. And to this aim, we just set up specific imaging protocols, including anatomical scans. So there was one 3D T2 weighted spin echo scan at a resolution of 100 micrometer. So the scan duration was 22 hours. And the second one, but a 2D T2 weighted spin echo sequence at 150 micrometer isotropic resolution. The scan duration was much shorter, 330. With a set also of diffusion scans, and the diffusion scans used a 3D segmented DPI post-gradient spin echo sequence at 3B values of 1500, 4500, and 80 seconds per millimeter squared along 25, 60, and 80 directions, targeting a resolution of 200 micrometer isotropic. And we use quite a lot of segments in order to discard any susceptibility artifacts or any distortion related to B0 inhomogeneities. And the scan duration was 82 hours. So in total, for each field of view, you acquire during 5 consecutive days. And here you have a picture of the 2 years acquisition campaign that represent around 6,000 hours of scanning on the 11.7 Tesla system. So we started in May 2018 for 1 year for the left hemisphere, and we did exactly the same in last year. And I would say that I could benefit from the fact that during the COVID party, I could get access to the 11.7 Tesla entirely. And this was done so in 2 years. In fact, it started in 2017, but we experimented the quench of the magnets, so we had to delay, the acquisition by approximately a year. And of course, there is a bunch of pre-processing to be done on the individual 11.7T scans. So to remember, for each field of view, we have a set of raw MRI data, including 2 anatomies and 176 diffusion-weighted volumes, amounting to approximately 8 gigabytes of memory per field of view. So we had first to correct for the noise using non-local means filtering of all the volumes. Then we had to compute mask from the highest anatomical acquisition, and then to compute a diffeomorphic registration to the MNI's nonlinear asymmetric space, which is our target frame to reconstruct the whole brain. And then we had to correct for the diffusion direction to construct the target volume, so that at the end you have a second times 8 gigabytes of corrected data for each of the field of view. This is an example of a corrected field of view. So as you can see, you have here the localization of the different field of view of the blocks. It was B1, B2, B3, B4. And you can see here the anatomical scan at 100 micrometer. You can see some really fine details. You have the same at 150 micrometers, where we don't see exactly the same details. So the combination of the two is quite interesting. And the first, volume at 0 seconds per millimeter square can be used also at a third anatomical scan at 200 micrometer. And you have here a rendering of the color encoded direction maps, where you can see very fine details about the superficial subcortical connectivity. So now, once we have done that for the 50 field of views, we have to do the reintegration of the field of views. So, as I mentioned, for each field of view of a given block, we have to compute the diffeomorphic transformation to the block scan at 3T. So here we have data scan at 11.70 matched to the scan at 3T. We do that for all the blocks, and then we need after to compute the diffeomorphic transformation from all the blocks to the block face we have acquired at 3T before cutting the brain. And ultimately, we have to compute a last diffeomorphic transformation to match, our block face image to the MNIs, to the ICBM 152, a nonlinear asymmetric 2009C MNI template. So in total, we had quite a bunch of diffeomorphic transformation to be computed, and it's useless to say that all those steps of preprocessing of restoration took a really long time because the data are huge. So you have to wait for the things to be done. And so we had to do that for the two modalities, the two anatomical scans and the 172 diffusion volumes. And at the end, what we've got is two reconstructed data sets. So one reconstructed data set at 100 micrometer isotropic. So just to show you, it's 2,400 voxels times 2,400 voxels times 1,600 voxels. We have the same thing for the anatomical scan at 150 micrometer, and we have the same thing for the 176 volumes of diffusion-weighted data. So the first anatomical scan amounts to 41 gigabytes. So as you can say, you have to use a strong computer to open and visualize your data set. For the moderate resolution at 250 micrometer, it's 12 gigabytes. And for the diffusion data set, it's 746 gigabytes of disk space occupied by the data set. So here I would like just to mention that, we had to revisit all the pipeline that we were used to use, because all the command line, they were not compatible with so huge data sets. And at the end, you can see here the kind of details here. That's the mesoscopic anatomical scan at 100 micrometer isotropic. You can see that you have some nice contrast in the cortical ribbon, and we also have some nice contrast, I mean, in the central deep nuclei area. So if we want to look into the anatomy, so I already showed you this picture. I mean, this picture, I mean, showed you the laminar structure of the cortex. We have some nice picture of the two hippocampi. And for instance, you have some nice details within, I mean, the deep nuclei that you can never see, I mean, at lower resolution. And as for the diffusion MRI data, you also have here, I mean, a nice picture representing 3 perpendicular slices of the color encoded orientation map stemming from the, I mean, the cubal orientation distribution function map. And as you can see some nice details. For instance, you see here the thalamic radiation that have some 90-degree sharp turns here. That was something that was hypothesized, but it was not able to efficiently see it, and with that resolution you can see it. So here are two other pictures where you can see that in fact you have quite a lot of details in the cortical ribbon. In the diffusion dataset, you can see some layers even in the diffusion data. I mean, those layers that just are tangential to the cortical, to the lamina, to the layers of the cortex. So that's very interesting. And we also have some nice details within the hippocampus. And I think we are now ready to start to look at precisely the structural connectivity of this human brain at the mesoscopic scale. So now, and I will finish with that, from the corrected data set, reconstructed data set, we are now proceeding with the segmentation of all the brain structures. So it will start soon. I mean, we will do the segmentation of the cortical ribbon, of the deep gray matter structures. And this task will be done semi-manually because we can use some existing software like SAMSEG, but we have to do some manual corrections afterwards. We also can compute the orientation distribution function maps with cubal or multi-shell, multi-tissue CSD models, that's spherical harmonics order 8. And we will get, I mean, we already did that. I mean, we got, I mean, an ODF map of 34 gigabytes. And we have also computed the tractograms from those ODF maps using quite conventional tractography algorithm and doing the filtering. And it, I mean, for just one seed per voxel, I mean, the size of the connectogram is 642 gigabytes of memory. So here again, you cannot visualize everything because, I mean, your GPU does not allow you to download such a huge amount of memory. And even so, with the processing, you have to devise some specific tractography methods, I mean, to be able to manage, I mean, those huge amount of connections. And here, I want to show you, I mean, that we have reconstructed the first 3D rendering. So here, because of GPU limitations, you only see 1% of the connection with only 1 seed per voxel. Let me tell you that in the future, we will reconstruct probably with 20 seeds per voxel. I mean, 1 seed per voxel is not enough, but here, even with 1% of the connection, you can look at those data, you can see the connection within the hippocampus, and without doing anything, you will see, I mean, the fibers entering the cortex, the cortical ribbon. They depict those sharp turns that were mentioned, but you cannot clearly see, I mean, at the millimeter resolution. And we are now computing, I mean, the connectivity matrix from those tractograms. So from the tractogram, you can do two kinds of things. The first kind of thing is to compute, to use fiber clustering techniques in order to regroup fibers into clusters and to regroup clusters to create white matter bundles. So that will happen now this year to design a Chenonceau white matter long and superficial mesoscopic bundle atlas. And you can compute also some structural connectivity matrices. And to do that, you have several strategies. Either you use the millimeter scale cortex parcellation provided by FreeSurfer. So that's the Desikian Cortex Atlas that Denis Rivière I put in the ICBM 152 nonlinear asymmetric space and the deep nuclei segmentation. Or we can also use the cortex parcellation stemming from the Hewlett brain, big brain atlas to have parcellation and I mean to study the connectivity based on cytoarchitectural parcellations. And we were asked by our HBP partner to compute a voxel-based connectivity matrix. And so even at the millimeter scale, we have more than 700,000 voxels. So we have to compute a matrix which size is, sorry, I did a mistake. So we will compute this millimeter scale matrix, but exploiting the data at the mesoscale, which will represent something like 100 gigabytes of disk space if we assume that 90% of the matrix is sparse. And if we wanted to do that at the full scale at 200 micrometer isotropic resolution, of course, the number of voxels is much higher, and it would represent 10 terabytes if we still assume that 90% of the matrix is sparse. Or we could also compute some parcel-based connectivity matrices. So as you can see, I mean, we are only, I mean, starting to exploit and to compute some interesting features. I mean, we compute those connectivity matrices because our colleagues from Marseille are developing a simulator which is called TVB, which would benefit from an enhanced connectivity matrix at the mesoscopic scale or at the millimeter scale that it will allow them, I mean, to do some simulation of epileptic events at a really fine resolution to see how, I mean, the electrical wave propagates over the cortical surface and within the hippocampus. We also have, just to finish, the possibility to compute proxies of tissue cytoarchitecture because we have acquired multiple shell diffusion MRI data. And because we have models that can provide proxies of the neural density and orientation dispersion, like the NODI model, we can decompose the signal, I mean, into the contribution of signals stemming from various compartments. So here I've represented a tissue which is corresponding to white matter, which will include the axons. It should be, that could also, sorry, the cortex that could also include some dendrites, some astrocytes, and some oligodendrocytes. And if we are just computing those models, we can get those kind of maps representing the neural density. So don't look at this non-linearity. It's just because this was an old scan and it will be removed soon. And we can also get, I mean, the orientation dispersion of the axon, which is also has a good contrast within the cortex. Okay, so we will be able to reconstruct all those maps of cytoarchitectural features in the coming future. And how can we use it? And that's something that was done. It is just an example done by Justine Beaujoin quite a long time ago. I mean, if you look at those cytoarchitectural features within the cortex, without doing nothing very specific, if you are just using clustering based on those set of parameters, you can recover a laminar structure where here, I mean, you can see some kind of things looking like layers of the cortex. And I would like still to highlight that we did not put any geometrical or spatial priors to get those maps. it was just computed without any geometrical priors. And we get something representing the layers of the cortex. And so at the end, we will do the same, I mean, for the entire Chenonceau brain. So now it's time to conclude. So the road was long and tortuous because we've experimented, I mean, a quench of the 11.70 MRI system. There was a pandemic situation in 2020, but well, we got there. So up to now, Chenonceau is the highest resolution diffusion MRI dataset available in the community, including both anatomy and diffusion MRI at 100 micrometer, 150 micrometer, and 200 micrometer isotropic resolution, respectively. We still have to publish it before lifting the embargo and make it available publicly to the community. And there is the construction of a mesoscopic Chenonceau MRI anatomical and connectivity atlas that will be soon performed in close collaboration with the iBrain team, in particular with the team of Christophe Destrieux. And I mean, there are now open discussions with HBP partners to start to establish joint projects in the frame of the last part of HBP. And of course, I mean, I'm also open to discuss some joint projects if people, I mean, in the audience are interested to exploit the data. I didn't mention it, but the last slide is here, and I would like to mention that, in fact, in parallel with this first session that was done during the 2 previous years, we also did a second equivalent acquisition campaign, also lasting 2 years, but this time on the preclinical 70 MRI system to collect further to collect further data for each field of view. And in particular, we wanted to collect T1 weighted variable flip angle SPGR dataset at 200 microisotropic, T2 weighted MSME at 200 micrometer isotropic, and T2 star weighted using a variable echo time SPGR, but with various echo times. And in fact, this will allow us not only to look at the cyto architecture, but also the Mielo architecture, because we are then able to compute, I mean, the quantitative T1, Yellin water fraction, T2 star, and T2 map, but also the iron load of the brain structure related to the T2 star map. So this second puzzle is currently under construction, and it will allow, for instance, to get that kind of data where here we can have a view of the lamina structure of the cortex, but from a Mielo architectural point of view. So we will be able to combine both the cyto architectural and Mielo architectural point of view over the cortical ribbon. And I think I'm done. I won't mention that. I just would like to acknowledge people that I mean, all the team with whom, I mean, the project was established. So Justine Beaujoin, who was the first PhD student, I mean, doing the proofs of concepts on the hippocampus and on the brainstem. Raissa Yegbat, who is working strongly on the Mielo architecture. Alexandros, who has developed the pipeline to reconstruct the Chenonceau brain and who is more focused on this connectivity and on the cyto architecture. Hivi, the engineer researcher of the team that has made a wonderful job to, I mean, to have all the pipelines revisited so that it works. Fabrice, who has developed this software that is used by Professor Destrieux to do the segmentation of all the structure of the brain. So here you have Christophe Destrieux, who is a neurosurgeon and neuroanatomist, who is, I mean, doing the manual segmentation. Of course, Fosy Boumesbe, who is the system manager of the 11.7 Tesla and who, I mean, helped us quite regularly when we had some problems. Jérémy Bernard, who designed, I mean, the containers to scan and reproducibly, I mean, the blocks, and François Geoffroy, who shared, I mean, his histological lab, who permitted, I mean, the fact that we wanted to cut the brain, to radiate, to etc., etc. And well, this project was funded both by ANR in the frame of the Fibre Atlas project and by HBP in the frame of this grant agreement. And I thank you, and I am open for questions.